Hello and good day. My name is Juan Manuel Martínez. I am from Mexico, studying at Universidad Autónoma de Nuevo León. And today I would like to talk about a research project denominated Design of a Multiprotope Vaccine for SARS-CoV-2, and its expression is along oligopersicum, a bioinformatic approach. Remember, in some points in late December 2019, the city of Wuhan in China alarmed the health system by reporting cases of pneumonia of unknown etiology. By January 2020, sample collection indicating an identity with other coronavirus reports in bats in China. In February, we already knew that this coronavirus was able to cause acute respiratory syndromes, and before March, the classification of SARS-CoV-2 for the viral agent and COVID-19 for the disease was already assigned. Currently, there are more than 34.3 million cases and about 1,002,000 deaths around the globe. The SARS-CoV-2 is an RNA virus that has a possible single-chain genome with approximately 30 kilobases, and 29 proteins have already been documented. The spike protein is known to bind the angiotensin-converting enzyme 2, for short AC2, receptor, allowing the virus to enter the cell, becoming one of the targets for the development of treatments and vaccines. Additionally, the distribution and expression of AC2 in different tissues in the body has been reported. These include respiratory and digestive system. The duodenum, the small intestine, gastrointestinal tract are included. At the protein level, AC2 is enriched in the digestive system based on RNA expression. With this information, we decide to apply reverse vaccinology, which is a bioinformatic approach that allows the prediction of macromolecules that are recognized by the immune system, also called epitopes. Through amino acid sequence of pathogenic microorganisms, it is possible to identify epitopes without having to culture them in the laboratory. Additionally, it is possible to produce these epitopes in plant expression system due to its security and scalability. To do this, we retrieve the amino acid sequence of the SARS-CoV-2 glycoprotein S, for short GPS, with the axis going as seen in the screen. The GPS topology was predicted with the CC-top served and confirmed with other tools. B-cell epitopes were predicted using the immunoepitope database and analysis tools, considering for the glycoprotein S antigenic, immunogenic, hydrophilic, accessible, flexible, and regions with beta turn. The identified epitopes were joined by means of helical linkage formed by 4 repetition of the following amino acid sequence each, and the models for the proteins in the structures were produced for the secondary, tertiary, and energy models. The best model was differentiated by analysis Ramachandran plot at Project. We also analyzed some features as physicochemical characteristics, solubility, allergenicity, and identity. To adapt and extend the fusion protein that we were building in the tomato, we optimized the nucleotide sequence of the glycoprotein by adding some genetic elements like cosette and sequel sequence at the sequence and result into a plasmid that we clone in Agrobacterium tumefaciens. The results showed that SARS-CoV-2 glycoprotein S sequence in the topology analysis server showed that the protein has 2 regions, one outside and one inside. Besides, 122 epitopes were predicted, but only 6 has critical characteristics. Due to previous investigation, the use of multiple antigens can increase vaccine effectiveness. This approach was chosen based on the previous research. The 6 peptides were locally located outside the protein, so they will be targeted by the antibodies. Helical linkers better control the distance and interference between the domains of the fusion protein. When we model the fusion protein with PDB served, these show a protein conformated for 84% of helices and 60% of other structures like holes and turns. The evaluation of the Ramachandran diagram showed that 96%, or approximately 350 residues of the fusion protein, were found in the most favorable regions. Besides, the analysis showed important characteristics like high solubility, non-allergenicity, high stability, and no identified in BLAST. The plasmid had a size of 50,478 base pair, including both the genetic elements and the fusion protein to be expressed in the tomato. The result obtained in this work suggests that the fusion protein design in silico as an edible vaccine has the potential to stimulate an immune response through the digestive system route to protect against SARS-CoV-2. In addition, the nucleotide sequence optimization will allow for the production of high levels of the fusion protein in the tomato plant. However, further in vitro and laboratory testing will be required to verify the effectiveness and safety of the vaccines. All the reference can be consulted at the end of the document. Thank you.